In this video, we are going to discuss about bacterial capsule. Okay, capsule surrounds bacteria as the outermost layer, so out of the cell wall. Okay, so when it diffuses into surrounding medium, it remains as undemarcated secretion. In case of leuconostoc, it is known as slime layer. So like it is loosely bound to the cell wall. So that time we call it as slime layer. Okay, and the capsule, which is sharply defined structure, okay, which is closely attached to the cell wall. So then we call it as capsule. So Streptococcus pneumoniae is one example of bacteria which is having capsule. Capsule which are thin and cannot be demonstrated under the light microscope. So the capsule is made up of polysaccharide. So function includes it enhances the virulence of the bacteria, inhibiting the phagocytosis, and it is protective against the antibacterial agent. In last video, we have seen that cell wall is the target site for antibiotic. Okay, so this capsule is attached to the cell wall, so it will enable the or it will stop the antibiotic by acting. Okay, so it acts as a protective covering against the antibacterial agents. And this capsular antigen we use for typing the bacteria. Typing means for the speciation or for the identification, we will use this capsular antigen. So example for capsulated bacteria includes Klebsiella pneumoniae, Haemophilus influenzae and Streptococcus pneumoniae. These three are most commonly isolated capsulated bacteria from the human sample. Okay, so we'll see how to demonstrate it. So usually we use India-ink preparation, also call it as negative staining, to see capsule under the microscope. Okay, so normal staining we will be not able to see the capsule clearly, so we go for India ink preparation, also call it as negative staining. So under the light microscope we can see. So here the ink cannot be penetrated inside the cell wall, or this capsule will repel the dye. Okay, so that's why the background will be stained here and your bacteria will be colorless. Okay, so that's why we call this staining technique as negative staining. Okay, then we have one more method, that is serological method. So capsular material will be mixed with the antisera. Anticapsular serum will be mixed and we will be observed for the swollen capsule. It is swollen because of increase in refractivity. So you can, in this diagram you can see the difference. So capsulated bacteria will mix with the antisera. So antisera, basically it is an antigen antibody reaction. So antisera, when we mix, it will act on the capsule, so it will be swollen, so we can observe under the microscope. Okay, so capsule is very important. In examination they may ask you. Okay, so this may be MCQ question, that the capsule is made up of polysaccharide, or the capsule is polysaccharide in nature. An example also important, that is Klebsiella, Haemophilus and Streptococcus pneumoniae. Hope you understood the concept of bacterial capsule. Okay, if you have any doubt, you can comment in the comment section. So I'll be happy to answer your question. Thank you.